We are having now a session on local adaptation to climate, with a focus on phenotypic traits that are important for trees to survive under changing climate. And I'm going to chair this session together with Eckert Benavides from the CSIC in Madrid, and I'm Santiago González Martínez from UAE in Bordeaux. And I'm going to introduce you to our invited speaker today, that is Andrew Eckert, coming from the Virginia Commonwealth University in the United States. And Andrew has done substantial work in population genomics and ecological genetics of forest trees, in particular conifers, and also in other species that are associated to forests, like some pests and different organisms. So welcome, Andrew. The floor is yours.
All right, thank you for the invitation. It's been wonderful so far. I hope to build upon the wonderful talks today with my talk, which is entitled Old Questions, New Data, and the Description of Local Adaptation. Before we sort of start building on some of the theory put forth by Antoine Crémer and all the empirical talks that were just amazing this morning, I want to remind us all about why we're here. We all love trees. Trees are amazing organisms that live in amazing places, that do amazing things, all right? And, you know, if we look at the places we get to go, these are all places that I've got to sample. It's amazing, right? The organisms that live there do amazing things. And I'd like to give an example, rather than some theory to start, with what I mean by trees are amazing or that they can be locally adapted. So if you look over here, here's a map of a very common North American tree species, lodgepole pine, Pinus contorta. And that very widely distributed species is broken up into a set of subspecies. And if you look on the colors over here on this map, each color is a different subspecies. There's 3 really common ones, okay, throughout the Rocky Mountains, the Sierra Nevada of California, and the sort of west coast. There's a 4th species that you can't see called Bolander pine, and it grows only right here on the coast of California in a couple spots. And where it grows is on a set of marine terraces that have been uplifted due to tectonic activity through time. The oldest terraces are up high, the youngest terraces are down low near the ocean. And on these terraces, they stratify 2 different subspecies of lodgepole pine. Shore pine on the coast, and this thing called Bolander pine up here on the upper terraces. They look dramatically different. Bolander pine is a pygmy pine. 100-year-old tree is this big. And it grows in this very distinctive soil that is a lot like serpentine soil. It's also flooded quite often. And it turns out that the differences in the phenotype between this pygmy pine and shore pine, which is a normal pine tree, grows real big, are genetically based. If you transplant them reciprocally, each one does better in their home environment. Still pygmy if you put it in nice soil. Okay? That's what we mean by local adaptation. And if you look at the distances between these lower terraces and upper terraces geographically, less than 5 kilometers. That level of phenotypic difference, 5 kilometers. Okay? If you want to talk about the genetic basis of this local adaptation, we did that with some candidate genes, and we turned out to find a root-expressed protein that limits the uptake of a certain ion in the soil that's enriched that causes pygmy stature. Yeah, there's a non-synonymous change that was almost fixed between these 2 subspecies. 5-kilometer differences. So that's my example of why we care. That's really cool to me. Okay? But those sorts of questions aren't new, as we all know. These are old questions. People have been wondering about the amazingness of trees for a long time. So, for example, we've been going out in natural stands collecting materials for centuries for a variety of reasons. Because they're cool, because we want to plant them, because we cut them down, we do things with them, right? We've also taken that material sampled in natural stands and put them in provenance trials and common gardens and all sorts of places that we do experiments with, right? There's so many of these that my graduate student went through and did a whole lit review of different ways of looking at quantitative genetic variation in trees. There's hundreds, almost thousands of studies with quantitative genetic estimates of traits in trees. This particular plot has different traits for a measure of trait differentiation called QST, and we'll talk a lot about that later on in the talk. Okay? The point of this is that none of this is a new way of thinking. That Bolander pine thing that got me excited, not new. We've been thinking about it for a long time, centuries. It's also a nice review of the sort of gene ecology for multiple centuries. All right. But those old questions now have new data. And the new data we've been talking about so far throughout the morning have sort of converged on genomic level data. Full genome sequences, or at least big chunks of genome resequenced, often annotated in genes, maybe non-genes, and you can compare and contrast, right? And those sort of new data allow us to reevaluate some of these questions. These questions about why trees are so amazing, how they vary across landscapes. We can put together those new data with these old questions. And we can even take those old questions and modify them a little bit so that the old questions become kind of old plus a little bit of new. Specifically, things about genome size have become very relevant for the way we think about adaptation. And we'll walk you through an argument put forth by Wenbin Mei and Jeff Rossabera that the way adaptation works genomically is a function of genome size. And they did this with two model plant species, maize and Arabidopsis. If you go through and look at the GWAS hits, so associations of phenotype to genotype, in Arabidopsis, they're primarily genic. They're in genes, about 3/4. If you do the same thing in maize, it's opposite. 3/4 of them are non-genic. They're actually intergenic. These are repeatable things, so they're not necessarily false positives. Okay? Some of them can be shown to have function that affects expression of genes. So again, these are true positives. So that's a very different way of thinking about adaptation, right? We want to think everything is about genes, but in the maize genome, which is about 6-ish to 12-ish times bigger than Arabidopsis, most of the functional variation that matters for phenotype is not genic. It's actually intergenic. Okay? Now, this also allows us to think about, well, why do we want to study trees? And why would somebody be crazy enough to study a conifer? And as we all know, conifers have enormously large genomes, roughly 20 to 30 gigabases in size. So why would you ever want to do that? Well, when we think about trees, we have a full spectrum of very small genomes, like poplar, half a gigabase or so, to very enormous-sized genomes that are not polyploids, conifers, pines in particular, upwards of 35 gigabases. We can compare and contrast various things about adaptation in these different genome-sized organisms. I'm going to concentrate today mostly on pines, just because I like pines, but I want to show you that, we can still do some interesting things even though the genomes are enormous. All right. So I'm going to do that with a set of 3 hypotheses, and those hypotheses will talk about how this relates to traits and adaptation. All right. The first hypothesis that I want to put out there is that populations of pines or trees in general are locally adapted at fine spatial scales. And what I mean by fine is within provenance. I'll go through that at length. The second hypothesis I want to put out there is that the genetic architecture, especially at these fine scales of local adaptation, is driven not by the fixation of alternate alleles across a gradient, but by subtle and coordinated allele frequency shifts. And we'll define what that means when we get to that hypothesis. And then lastly, I want to extend it out a bit more and talk about how adaptation and introgression from hybridizing species can interact to produce fitness differences. Specifically, that speciation within Pinus is often associated with gene flow across the divergence history that can be thought of as maybe continuous, and extrinsic selection upon introgression that does happen with that gene flow. And by extrinsic selection, I mean selection due to environment of the admixed or hybridized individuals. All right, so let's go to the first hypothesis. As we move through the talk, the hypothesis is listed up there, so if you get tired of me yakking on, you can kind of remember what we're on. There were 3. This is the beginning of 1. All right, so when we think of local adaptation in trees and what has been done mostly, we think about this sort of standard setup of a range-wide provenance study, where we take our organism of interest. This is white bark pine. It grows in the western sort of portions of North America. Very iconic, high-elevation species. We go through the range and we collect materials from sites in that range. We establish them in a common garden or set of common gardens. and we do some quantitative genetics. Right? That's the typical way we think about this, right? And one of the primary things we do with those quantitative genetic estimates is calculate this thing called QST. It's a measure of trait differentiation, the genetic basis of the trait differentiation. More specifically, it's the partitioning of the additive genetic variance of that trait variation due to among sites. Okay? So the bigger QST is, the more differences there are across the sites in your study for the additive genetic variance. Okay? So if you do this in white bark pine, like Sally Aiken and Andy Bauer did in the, I think it was early 2000s, you calculate this measure of QST, you can get estimates, it ranges from 0 to 1, upwards of 70% for some traits. That's bud flush in a certain year. Okay? 70% of the additive genetic variance for that particular trait in that particular year of the individuals in the common garden due to differences among the sites from which the materials were collected. That's huge. If you then compare that to the sort of differentiation you'd expect from molecular markers, we call that FST. It's calculated very similarly. It's not additive genetic variance anymore. It's variance in allele frequencies across sites at markers. That's that dashed line right there. There's QST, there's FST. From that comparison and some fancy statistics, you can conclude that, hey, this trait is way too differentiated relative to molecular markers, so that whatever QTL underlie the trait have too much differentiation. Local adaptation. But note that we did this range-wide. And the prevailing hypothesis is that range-wide stuff is very evident of local adaptation because you can get these big sort of gradients across latitude, strong selection pressures in the south relative to the north because there's so much variation. And if you look across species, we find this pattern as Bauer and Aitken did quite often. About 3/4 of the time that this is done range-wide, you get QST much larger than FST in trees. Lots of local adaptation. But what I want to ask is that if I don't do it range-wide, and I go to something that we would call a provenance, a particular site range-wide, the Lake Tahoe Basin of California and Nevada, and collect at local sites within there in a fully quantitative genetic design, do I see similar patterns? And by local, I mean, here's Lake Tahoe. Each of these dots is a collection site from which we collect multiple families. We establish them in a common garden. We have many offspring per family, roughly 25 to 30, and we do the full quantitative genetic analysis. We did this for 3 species: sugar pine, western white pine, and white bark pine. They're stratified elevationally in this basin. Here's a picture of our common garden. In this common garden, we had a randomized block design. We had growth and survival-related traits: bud flush, growth, carbon isotope discrimination, nitrogen content of needles, root tissue biomass. We took GIS-derived data about the origin of these maternal trees, the sites from which they came, and then we estimated values for each family and how much of the additive genetic variance was due to site using linear mixed models and the variance components from it. So let's start with trait variation for each of the 3 species. There's 2 traits shown here. We had 5 in total. Here is height growth. Here is carbon isotope discrimination. Each point on here is a site. Each set of gray bars is a different species. The gray bars are the global mean plus the 95% confidence around that global mean. Each point around that gray bar is a particular site for that species. Its error bar is the variance across families in that site conditioned on the global estimate. And what we see is that we see variation everywhere. We see variation across the species. Let's take height growth. That's sugar pine. That's western white pine. That's white bark pine. So the bars don't line up. We also see variation around the global mean. We see variation across sites relative to the global mean. Variation is everywhere. If we concentrate just on within site, we can use this framework to calculate our standard measure of evolvability. It's debatable whether it really is one or not, but heritability, narrow sense heritability. And here they are by species for the 5 different traits: sugar pine, western white pine, white bark pine. We see substantial heritabilities for most traits for each species. We also see differences among the species. Let's take height growth, for example. Sugar pine has a heritability for height growth of about 0.35. In white bark pine, it's close to 0. It's about 0.08. All right? So we can see variation on that level. If we want to talk about variation among sites, QST, using the same framework, here they are. Shown to you for the three species, sugar pine, western white pine, white bark pine, are estimates of QST from those linear mixed models. Those shown in red are statistically different from 0, meaning the additive genetic variance is not 0 partitioned across sites. So there's some effect of sites for at least one trait for each species. Okay? So QST is not 0 for everything that's in red. And you notice that sugar pine and western white pine, 4 of the 5 have that true. For white bark pine, only 1. So there's differences on that level. And then lastly, we can take that and relate it to environment, that's shown on the left here, as well as do formal tests about QST being larger than FST. So let's go over to the environment. Shown here are redundancy analysis for environment predicting traits. And that environment was stratified into climate, soil, and representations of geography through Moran eigenvector maps. It's just a decomposition of a pairwise distance matrix where the distances are kilometers. We can talk more about that in the end. What we see is that those three things that we call environment can explain upwards of 50% jointly of the trait variation. That's huge, significantly so. Moreover, using this approach, we can decompose pure effects of climate, soil, and geography, combining to do that 50% explanation. That's these bars over here. Right? Climate and soil have different effects across species. Soil is often as important or more important than climate in predicting these trait values, specifically the genetic values of these traits. If we then test whether or not the QST from the previous table is bigger than FST, using the method of Whitlock and Guillaume, which takes into account the substantial variances of these things, anything in blue over here is bigger than what's in red, which is FST. Blue and gray are QSTs, that's FST. Anything blue across species is significantly bigger. So what can we conclude from that? Well, we can conclude that within provenance level variation exists, genetic variation. But if we just said, hey, this is a provenance, and we lumped everything together, we're masking lots of signal of local adaptation. That variation is consistent with local adaptation, and it varies by species, even in the same geographic area, even when the populations are like, on average, 5 to 6 kilometers apart, which is about what the average was for those. What that means more broadly is that gene flow, or the potential for gene flow, lots of it, does not preclude the development of local adaptation. That we can't just say, hey, FST is low, gene flow is high, therefore no local adaptation. And we can say that for a variety of reasons, the data I just showed you, as well as, moving on to hypothesis 2, some wonderful theory that Martin Lescaut mentioned this morning that Antoine Crémer and colleagues came up with. It's related to the genetic architecture of the traits under selection. So that for some architectures, we expect not a lot of differentiation at the loci that cause the trait itself. Right? Specifically, architectures of large numbers of loci with small effects. The FST for any given one of those loci is going to be small and not unlike the average marker in the genome. What matters in those cases then is not the allele frequency differences at the QTL that determines the trait, but the covariances of how those loci develop across populations, as well as within populations. So you can sort of think about it this way. QST can be written as a function of the FST of the causative loci, that's FSTQ. You can also sort of decompose the covariances into between populations, within populations. What I want you to look at is the numerator here, that this is the covariance of allele frequencies or allelic effects across populations, the relative proportion of it. It amplifies the FST at any given marker causing a trait. So that QST can be big because that can be small, but that big, or it can be big because that's big, and that's big, or just that's big. Okay? Keep that in mind. So what we did is we tested this hypothesis with one of those three species, white bark pine. Same samples from before, but now we went through and genotyped the heck out of them using an approach called DD-RADseq. Okay? And talk to you at length about the difficulties of ddRADseq in conifer genomes, but it does work fairly well for this purpose. What we did with the genotypes for those data is we did a genome-wide association study, G2P, genotypes to phenotypes. We also did an environmental association study, genotypes to environment. Those are the two approaches we use. That's a Bayesian sparse linear mixed model version. It's a sort of multi-locus GWAS approach. That's a single locus approach developed by Graham Koop and Torstein Günther. Then we asked, given the loci identified in those two approaches, do they display a particular form of allelic covariance of effects? Are the allele frequency differences across populations covarying more so than random markers? And that's the little equation there. This is statistical linkage disequilibrium. So unlike Tanya's wonderful talk from before where there was a massive haplotype and an inversion, this is not that. This is covariation of allele frequency change in certain ways across populations. So what did we find? Well, we found dozens to hundreds of loci for each of these approaches by trait as well as by environmental gradient, using those two association approaches from before. Shown here are some hyperparameter estimates of the sort of SNP-CHIP heritability of the causative loci. We can talk at length about that. This is the number of loci in those models, well over 100 in those models predicting each trait. Lots of causative loci. If we take those causative loci here and in the environmental gradient, we ask the simple question, does FST differentiate loci that are interesting from this perspective or that perspective? The answer is no, not very much. The average FST is at best a tiny bit higher for the causative loci relative to random markers. That's the causative locus set. That's the same sort of null distribution of the same number of markers matched on heterozygosity, FST at single loci, and missing data. Okay. Those are loci that seem to cause, this is for, I forget which trait, one of the phenotypes, okay, relative to random markers. Okay. The sort of environmental associations are a little bit more structured, but not much so. And in fact, if you look at single loci, their FST values fall right in there. This multi-locus FST is elevated for the reason I'm going to show you on the next slide. And that is, these allelic covariances differentiate these causative SNPs more so than random SNPs. It's not FST. It's not anything else about raw allele frequency differences. It's these covariances. This is exactly what's predicted by that theta B term being more important from that equation from before. Shown here are each of the traits for white bark pine. Here is the median level of pairwise among population covariance and allele frequency. That's the white bar. The black bar is the null distribution. It's the 99.9th percentile of the null distribution of medians for random sets of SNPs matched on all those criteria. And what you see is that the median of the observed value is often larger, and often quite larger, than the 99.9th percentile of medians in the null distribution. Pretty high. And it's even more exaggerated for the environmental data. So all of those causative loci are not differentiated by FST outlierness or allele frequency differences, but these covariances. What can we conclude from that? Well, we can conclude that local adaptation, at least in white bark pine in this system at fine spatial scales, is driven by subtle and coordinated allele frequency differences. Like the allele frequency differences are less than 5% different for these loci relative to random genes. But they're correlated to traits and they're correlated to environments very strongly so. These SNPs don't look different much from random SNPs as far as those allele frequency differences. The environmental associations did have stronger signals of selection. You noticed that those covariances were much larger observed than the black bars. Okay. If you sort of decompose further, which traits and which variables show the biggest differences amongst the observed values relative to the expected values, there were things related to water availability. And in fact, if you remember in any of those QST values we whizzed through, the only QST that was significantly different than 0, as well as significantly greater than FST for white bark pine, was something related to water use efficiency, carbon isotope discrimination. The environmental data are consistent with that as well. All right. So how would this all play out to systems of conifers where we don't just have one species distributed across a gradient, but you might have two interacting species distributed across a gradient? Specifically, two closely related species that are maybe separated on a latitudinal gradient, south to north. Okay? What I'm going to do is use that idea to test this hypothesis, that speciation within Pinus is often associated with continuous gene flow and extrinsic selection upon introgressed alleles, given that sort of gene flow between lineages. And there's been a long history of studying reproductive isolation in conifers to varying levels of success. Here is some work of Bill Critchfield showing crossability amongst members of the clade strobus in Pinus. Each line connecting a dot is the ability to cross members manually. Like he could put pollen on the female cone and get a seed that germinates in the greenhouse. Often, these patterns of reproductive isolation and or ability to hybridize can be thought of as contributing novel variants to the process of speciation. There's a well-worked-out example of homoploid hybrid speciation, where the sort of hybrids of a cross evolved along a novel environmental space into their own unique species. It's 3 species of pines in China. All right, so those are not the species I'm going to talk about. I'm going to talk about 2 species that I've been working on with this very talented person who's a graduate student in my lab, Mitra Menon, and that is southwestern white pine and limber pine. These are distributed on a south to north environmental gradient. In the south, you have southwestern white pine, goes into Mexico, as well as the desert southwest. And up in here and north, you have limber pine, different species. In fact, these could be sister species, but if they're not sister species, they're really closely related. These 2 species have different climate niches. And if you break these 2 species, this southwestern white pine, into, hey, there's a disjunction right there, and you look at these separate from those, which is supported by the genetics, they themselves have a different climate niche here relative to there, which is different than here relative to there. So we have hybridization happening. We have a clinal pattern of allele frequencies. We have niche differentiation of things that appear to be in a zone where hybridization is happening. Okay? If we fit a demographic model using some fancy method like diffusion approximation of demographic models, DADDY, we get a model of divergence with gene flow as the best fit model, and that gene flow has been happening continuously since divergence. If you then use that model and that thinking to identify loci that have weird patterns of integration, you can find outlier loci that retain too much ancestry from one of the parent species. And that's this genomic Klein analysis. There's a set of loci that have too much ancestry from strobiformis, which is southwestern white pine, relative to the genome-wide average expectation. We have a different set where it's the opposite, too much Pinus flexilis or limber pine ancestry relative to expectations genome-wide. If you look at those, the outliers that have too much flexilis ancestry are correlated to certain environmental variables, cold, temperature-related variables. Those that have too much ancestry for southwestern white pine are correlated to a different, exclusive set of variables, drought-related variables, water availability. These 2, cold temperatures and drought availability, also drive the differences in the niches. So that's pretty cool. Okay. So we stopped and said, hey, that's pretty neat. Can we replicate that with a new set of data where we solely sample the putative hybrid zone, that little zone in the southwestern United States? Not just the Mexican ones, not just limber pine, but we went out and sampled 110 locations in the hybrid zone, in a paired transect level design. It's roughly 1,100 trees, generated a new SNP data set, and we did the analysis we did before, plus an environmental association analysis. And we compared and contrasted those 2 sets of data for the loci that were interesting. All right, so let's go through this graphic right here. If we put on this axis the environmental variables that are most different between the species, those are mostly the cold temperature-related variables. What we see is that introgression outliers that I talked about before are enriched for signals of local adaptation when you consider just the hybrid zone from an environmental association analysis. So much so that if you just looked at the hybrid zone by itself, you would conclude local adaptation. And you would come up with some explanation about standing variation contributing to local adaptation. But what's driving this is not standing variation. It's introgressed variants from Pinus flexilis related to cold. It's those same variants from before that have excessive levels of linkage disequilibrium amongst them that are enriched for driving those signals, significantly so. Okay, so that's pretty cool. And in fact, when you take that hybrid zone and you look at, hey, where there's lots of admixture, you see, and introgression, you see those populations expanding northward demographically. So those hybrids now have a different demographic history because of those alleles. They're able to go to areas that are cold, where the ones that don't have those alleles can't. Okay? And that's sort of evidenced by comparing and contrasting Klein centers from phenotype and genotype. Okay? So what can we conclude from that? Well, that exogenous selection affects genetic diversity generated through hybridization and introgression. There were Bay and V outliers in the data set that we defined as hybrids. Different parental groups provide variation associated with different environmental gradients. Drought comes from southwestern white pine ancestry tolerance. Cold tolerance comes from the other species. And that if we just look for signals of adaptation, we can mistake the cause or mechanism if we don't consider other things like introgression. All right, so how did this generally inform us? Well, we concluded that genetic variation, at least in the system I talked about, is structured at fine spatial scales. That begs the question, well, how fine of scale matters for conservation and management? Well, I can't give you a prescription. I can at least raise that question because I've shown you that assuming that sort of provenance level variation is just all kind of the same due to gene flow is a bad idea, at least for some systems. We also showed that different traits in different species give conflicting signals of adaptive evolution. QST was bigger or smaller depending on the species for the same trait, measured at the same age. That begs the question, which traits should we measure and manage? And then lastly, if allelic covariances are the driver of adaptation, if you calculate those relative to the single locus variants, they were as big as those summed for the pairwise comparisons. How do we manage for allelic covariances? I don't know how to do that very well. Maybe I'm just slow or something, I don't know. But I don't know how to do that well because I can't just quantify allele frequencies and pick. All right? Well, how could you extend this further? This is the end of my talk. I might finish on time. Okay. There are three sort of areas I think that we could move forward with. The first, which traits affect fitness? And are there multivariate interactions among traits? How do these change across the ontogeny? And what I mean by that is here is the pairwise correlations in a network fashion of the traits measured in the common garden from before. Right? Each of the lines is a correlation coefficient of the genetic values across families. Stars designate significance. Red designates a negative relationship. Blue designates positive. That's sugar pine, that's western white pine, that's white bark pine. There are significant correlations across traits that I ignored when I did all the quantitative genetics and QST, FST outliers. That variation also varies across species. Those correlation matrices are significantly different from one another at the same age across species. If I change the age, do these change? In fact, they do. So I have some data for western white pine that shows that they do. Okay. And then another area of thinking about is that everything I did was in a common garden. It allows precise estimation of variance components, controlling for the environment of the garden. But does that really translate to that, a natural stand? And if so, how much? Right? What I should be doing is some quantitative genetics and natural stands like Antoine, and then relating it to common gardens. Or conversely, should I be designing samplings in nature to do an experiment in a common garden, rather than just picking randomly? And then lastly, what are the evolutionary patterns of adaptation, both traits and genes, across different clades of trees? Some seminal work by Sam Yeaman and colleagues talked a lot about convergent evolution, the genetic basis of it in spruces and pines. That sort of gets rid of the phylogeny because it was so long ago, it's not anything to do with parallel changes. What I'm talking about is taking a clade of closely related species and figuring out how traits and their architectures evolve on that particular phylogeny. Good old comparative phylogenetic type stuff. That's not done in trees very much and can be informative. None of this would have been possible without all these super talented people here, collaborators folks in my lab, funding agencies listed over here. Thank you.
Thank you so much, Andrew. Do you have questions? Yeah.
So, do you have insights about the covariance? Like, you use the association with the phenotype or environment, but when you don't know, you don't have the association results, or you don't know the traits that are adaptive, or you don't know the environment, how do you tackle this problem when you have like million SNP?
Yeah, so that's a good question, Tanya. Thank you. So I was able to define sets of loci to investigate as, hey, this set is interesting for some reason. Is it further interesting in a different way? If we don't have the ability to make sets, I don't have a good method for you off the top of my head. One could imagine a computational problem where you tried to make random sets of things, maximizing some measure of what you expect in some way, but the computation to do that would be enormous with 1 million SNPs. This is all based on pairwise interactions among SNPs across populations. Some computer scientists may be able to figure that out, though, the optimal way to do that in some kind of approximate way. So you need sets of SNPs. That's the weakness of doing this. And preferably sets of SNPs defined independent of the measure of linkage disequilibrium, so it's circular.
Hello, very nice. Thank you for the talk. What's your opinion about epigenetics? When these very short-term common garden experiments, do you really think that you can be biased by that, or just?
Right. So the question is, how biased are the estimates in a common garden from epigenetics? We know that maternal effects are important. We know that if temperatures are a certain way during germination, then the offspring in a garden will be different than if that wasn't there. That's been shown in spruce, lots of different things. Right? That is truly important. And one way to think about maybe maternal effects is to include covariates for the estimation of values, such as seed weight, seed size, temperature during collection. Often, if we can do it, we'll provide some measure of at least seed size to regress out any effect due to larger seeds across moms making different sized seedlings. Right? That's one way to deal with it. Okay, we know it's important. If we don't do that, then we have to wait longer for the trees to get older, beyond a year or two. And even then, it may take an unknown amount of time to blur the effect of that.
Sean Hoban, Morton Arboretum. Your first example took place in Lake Tahoe, which is a really extreme environmental change over a super short distance. Yes. I think you can ski at the top at the same day you can swim at the bottom. Correct. So, but large parts of the US and Europe and globally are really homogeneous over space. So do you think those conclusions about conservation management at such small scale are broadly applicable?
Right. So that's an excellent point, and I brushed that under the rug, assuming that you didn't know that. So this sort of thinking applies very well for very strongly heterogeneous environments that change across space. But where I'm from in Virginia, you can drive 100 kilometers north, and it's not that different than where I just was, right? Unless I go up in elevation, which is not a lot of that in general. And so that conclusion that fine spatial scale matters is conditioned on some source of selection varying across those scales. Right? And so that could change as you get different landscapes. That is true. How much it changes, I can't quantify because I didn't do a comparative study. But you would expect it to probably, and if you don't expect it, you could at least test it with a comparative analysis, which should be done.
That's just a technical question. The covariances are calculated only with the allelic frequencies, also with the allelic effects, with both, yes. Right?
So the measure that I provided was of the allele frequencies only, because I wanted to compare and contrast environmental associations, which don't have allelic effects in the same way as the traits. I could turn them into allelic effects using the standard conversion that is based on that plus the effects of each of the loci, right? And if you do that, the traits do show the same sort of patterns. Right? So this is the covariance and allele frequency change, not of allelic effects, but that forms the basis if you include the allelic effects. Okay?
Thank you so much.